Hello, everyone, and welcome to today's webinar, Tools to Empower Your NK Cell Research. I am Michelle Ashton of Labroots, and I'll be your moderator for today's event. Today's educational web seminar is presented by Labroots and brought to you by Miltenyi Biotec. To learn more about our sponsor, please visit www.miltenybiotec.com. Let's get started. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask the Question box and click Send. If you have trouble hearing or seeing the presentation, click on the Support tab found at the top right of the presentation window, or report your problem by clicking on the Ask a Question box located on the far left of your screen. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located at the top right of the presentation window and follow the process to obtain your credits. I now present today's speaker, Martha Luevano, the Product Manager of Research and Clinical NK Cell Products at Miltenyi Biotec. For a complete biography on our speaker, please visit the Biography tab at the top of your screen. Martha, you may now begin your presentation.
Welcome, everyone. Thank you so much for the introduction. I would like, first of all, to thank you for attending this webinar. My name is Martha Luevano, and today I want to share with you how to drive your NK cell research further. Whether you're doing basic or translational research, I am sure that you will benefit from this webinar. So let's start. First, this is just an important disclaimer that states that unless otherwise specifically indicated, Miltenyi Biotec products and services are for research use only and not for therapeutic or diagnostic use. So just please acknowledge. So today, we will navigate the different technologies needed for a successful NK cell research, from cell separation to cell analysis and going beyond to translation of all your findings. Let's start with a brief introduction. I would like to just highlight that Miltenyi Biotec has been a key player in the field of immunology for now 30 years. Our products have been published in more than 20,000 scientific papers, and we have invested a lot in R&D and have enabled cell therapy procedures all around the world. Now let's get into the NK cells, my favorite immune cell type. They play a very important and key role to fight tumors and virally infected cells. They are able to kill a variety of targets through a contact-dependent, non-phagocytic process that does not require prior sensitization to an antigen. And this is what makes NK cells really useful and wonderful, this part of no sensitization to an antigen. And how are they able to kill the target cells? NK cells are able to use different mechanisms, including direct lysis of target cells via perforin and granzyme B, also by secreting different cytokines, for example, interferon gamma and tumor necrosis alpha, or by inducing apoptosis using fast ligand or TRAIL. In addition, NK cells are able to recognize antibodies that are already bound to target cells in a mechanism called antibody-dependent cell-mediated cytotoxicity. And this mechanism is used by a variety of monoclonal therapeutic antibodies that are already in the market. So how can we exploit NK cells in immunotherapy? Well, NK cells can be obtained from healthy individuals. They can be isolated, expanded, and infused into patients suffering a wide variety of diseases that are now under investigation. Moreover, with the promising results of CAR T cells, NK cells can also be genetically modified, offering several advantages compared to the T cells. First of all, you can observe here that you can use allogenic NK cells from a healthy donor. And this is one of the main advantages. However, there are also more, including better killing or also using all these different mechanisms of killing that I explained to you before. But let's now dive into another section of how you're going to study your NK cells. And the first part, of course, will be to isolate your NK cells. This is the first step that you will have to encounter when you're studying your NK cells. So I would like to have this slide here because we get a lot of questions regarding what is the benefit of the column-based technology in comparison to our column-free system. So I will spend some time here because I think it's important for you to also have it in mind. The magnetic separations in column-based and column-free system have in general a very comparable magnetic force. However, the difference between the two systems is, of course, the column. The columns are filled with little iron spheres, and these iron spheres, they pass through a magnetic force on the next over a very small distance, amplifying the magnetic force about 10,000 fold and ensuring that the magnetic field is constant throughout the column. Thus, minimal labelings of cells with these nanobits is sufficient to retain any target cell that you label. If you look at the picture on the right-hand side, you see a comparison of a cell labeled with bigger beads using a column-free system on the left and our tiny microbeads on the right. And you can also see in the electron microscopy pictures in the bottom that the bigger left stick to the surface of a cell. In this case, it's a T cell, and this could lead to activation and alteration of the cell characteristics. Moreover, in our column-based system, the cells can run freely throughout the column and can be washed thoroughly to remove cell debris and also contaminants. Finally, you can always take the advantage of thinking translational because these microbeads have also been used all over the world in clinical trials. So what options do you have to study and isolate NK cells? On the one side, I will just give you an overview. The traditional approach, you will go from your blood sample to produce PBMCs via density-graded centrifugation. NK cells then can be isolated using max microbeads, that will be a positive selection, or max isolation kits, that will be negative isolation, meaning that you remove every single cell that you are not interested in and leave your target cell untouched. Next, you can also have a straight from technology. In this case, instead of doing the density centrifugation, you can use our new straight from technology. NK and NKT cells can be directly isolated from different blood products in fast and easy way without the PBMC isolation step. In this case, we are using CD56 microbeads. Also, if you're looking for an untouched solution from blood, you can use Max Express, and NK cells will be untouched. I just want to highlight some of the kits that we have. If you're not familiar with them, we have available CD56 microbeads and also our new release CD56 microbeads. In case of isolation kits of untouched cells, we have a couple of kits, but the most useful one is the NK cell isolation kit. And here I just want to give you a little sample of what does it look like using the release. So this is a new product that we just have released last year. And in this case, from PBMCs, you can use a positive isolation, so CD56, and then if you want to, you can get rid of the microbead and also the antibody attached to the cell. That means that you can use the cell for another magnetic separation. And what is important here, because I have heard a lot of fears regarding using positive isolation, you don't have to worry about cell activation. You can see in the graph before separation of the PBMCs, the activation markers on the lower row, CD69 and CD25, and you can see that there is a very little amount of percentage activated cells. After enrichment using the CD56 microbeads, this percentage does not change too much, and after depleting the CD3 cells, also you get just a very pure NK cell product without activation. So this is a really great tool, especially if you want to subsequently label for other type of cells, for example, an NKG2C positive cells or KIR2DL positive cells. The second option, of course, you can start from different blood products and skip the density gradient step. And for that, you can use either whole blood or buffy-coats or LRC, and they all have different amounts of erythrocytes and, of course, PBMCs. And we have developed a specific straight from microbeads for each of these products. In this case, I show you here just an example of what traditionally you are doing. So I don't go into details. You are probably very familiar with it. Once you get your sample, you have to do your gradient density, your centrifugations, your PBMC washes, your counting, and then you start your isolation. But comparison to that, if you use our straight from technology, you can easily start without even counting cells with your buffy coats, for example. You add your microbeads, you incubate 15 minutes, and with the Multimax 24, you can easily get in less than 5 minutes all your cells. Most importantly, you have high recovery, good results, and no cell activation. And here in this slide, I would like to show you how does it look like. If you're using our CD56 microbeads, because there are no washes involved, you have a really great recovery and really nice purity. Just remember that whenever you're using CD56 microbeads, you will have a mixed population of NK and NKT cells, as you can see in the plot on the bottom right. Okay, now let's continue to the other great challenge of culturing NK cells. In the workflow, I will show you just as an example. I have written down here step-by-step generation of CAR NK cells, but this workflow can be used with or without the CAR. So in general, if you are going to culture NK cells, this is information very useful for you. I believe that you are probably very familiar with these statements. So when you try to expand NK cells, they really don't like it because they easily get exhausted. And most of the time, you will find that NK cells do not retain their cytotoxicity activity after expansion. And this is a problem, especially when you're studying the NK cell functionality. So what we have developed for these reasons is a specific NK media. This media has proven to have superior expansion, and I will show you in the next slide. It's free of any animal-derived components. It really limits the growth of unwanted cells, like B cells or T or D cells. And most importantly, it is available up to max GMP quality. So let's look at the data. Let's say that you start with NK cells, isolated NK cells, and you want to expand them. If you're using our media, you can get up to 5 times more NK cells compared to our T cell media, the TexMax, or other competitors that are really not NK media. They are mainly formulated for other type of cells like stem cells. So this is a really nice data, and I have received a lot of feedback from customers saying that they get also very similar results. In the next slide, if you are not starting with isolated NK cells but with PBMCs, even starting with this population, you will find on the left side that you get around 3 times more NK cells compared to other media. And the most important thing is, if you're using our media, the majority of the cells at the end will be NK cells. In the graph on the right, you can see that around 65 to 75% of the cells are NK cells when using our media, compared to 35 to 40 if you are using other media. This is a protocol using interleukin-2. If you combine interleukin-2 plus interleukin-15, you actually get higher rates and more NK cells. So in reality, what is very advantageous from this media is that you can do everything, all of your experiments in small scale, in research grade, so NK Max medium research grade, and when you find the optimal protocol, if you need to translate this to the clinic, it's so much easier because we have available the GMP medium that has exactly the same formulation as the research version. And here I show you some data. If you're comparing a research version and a GMP on the left side, you find very similar fold expansions. And the most important thing that I mentioned in the beginning, NK cells preserve the cytotoxicity activity after expansion. So after 14 days of expansion, we tested the cells against cell line K562. This is a standard 4-hour killing assay. And you can see that the killing was absolutely preserved. And it goes for both, for the research and also for the GMP medium. So of course the medium is very important for your experiments, but also you will find out that you need to have good cytokines. One of the biggest issues using the cytokines is this variability between different lots. And to overcome this, you could in theory perform this lot-to-lot testing, but why should you do that and waste your time and money in something that we have done for you? So with our premium cytokines, you can rely a lot on the lot-specific activity value that we provide you in a certificate of analysis. Using the cytokines, you do not need to do any pre-testing, which will save a lot of time and money, and you can apply the same amount of active cytokine in each and every experiment for maximum reproducibility. So these human-grade cytokines share the same characteristics as the max GMP cytokines, like the low endotoxin content, high purity, and they will be very relevant for your preclinical studies. Again, they are also in GMP quality, so your translation will be really fast and easy. So, of course, if you're going or thinking about CAR NK cells, you might wonder what tools do we have to help you. Well, NK cells are definitely a difficult cell type to transduce. Normally, it's very often that the transduction is inefficient and you require the use of something to enhance this transduction. We have developed Vectofuscin-1. This is our transduction enhancer, and it was developed to enhance viral transduction of cells growing in suspension like T cells or NK cells. Chemically, it is a fully synthetic soluble peptide that is non-toxic, enabling these higher transduction levels with low amounts of retroviral or lentiviral vector. It is very easy to use. You just premix your virus with the Vectofuscin, incubate 10 minutes, and then just add to the mixture of your NK cells. The most important thing here also, you can have this reagent in GMP quality when you translate this into your large-scale manufacturing. If you want to see some data, here is some. So these are primary CAR NK cells. On the left side, you will see non-transduced, then only the vector, around 70% of the efficacy. And if you use Vectofuscin, then this enhancement is up to 85%. So this could be really valuable in your research. I want to show you some data from customers. So this enhancement of the transduction efficacy depends, of course, on your virus, but definitely if you have using our media and our Vectofuscin, you have more chances of getting more CAR NK cells. This is customer data, so they try their own systems or their own media with retronectin, and I gave them some samples of Vectofuscin and max media. On the left side, you can find that the NK cells using our media were grown even more, so around 60-fold expansion or 60 million, compared to the competitor, which was around half of them. So in this case, Vectofuscin kept the same transduction rate, around 50%, but at the end, you will have double the amount of CAR NK cells if you use our system. So again, I think you have to just test it, and it will depend on your virus and your protocol, but I am almost sure you will see a benefit. Now let's go to the part of cell analysis. This part of the workflow must be the most important because you have invested so much time and so much work, and you need to make sure that your analyses are right. So let's start here. If you know something about Miltenyi Biotec, it's probably cell separation, but I just want to show you that we have so many tools for your flow analysis. We really offer a comprehensive product solutions for this flow analysis, not only from antibodies and dyes, but we also have ready-to-use kits and assays so you can measure all of your samples and get the best data from it. If you're not familiar with our recombinant antibodies, I just explained to you really quick. If you're trying to do this in-process control or quality control, or even just study your NK cells, or reaffinity antibodies are perfect for you. They are recombinantly generated and ensures very high purity and very high lot-to-lot consistency for greater reproducibility. And all the different antibodies have the same universal IgG1 isotype that leads to the fact that you only need one isotype control. That would save you some effort and cost, and also the complexity of your experiment planning. And last but not least, we have the re-antibodies are characterized by a mutated Fc region. That means that you can eliminate the Fc receptor blocking step. There will be no unspecific binding. If you want to know more about our recombinant antibodies, please have a look in our webpage. We have a webinar giving you all the information that you need. So, okay, now you know that we have antibodies, but you're also wondering how can you study your NK cells in terms of functionality using cell analysis. So we have here just an example of two different assays. The first one is cytokine secretion assay, and another one is a standard killing assay. Both of them can be performed using flow cytometry, and I will show you how. Probably you would think of cytokine secretion assay more like an ELISA or something that does not require flow, but I want to show you a very nice tool on how to analyze all of these cytokines at once. So this is our actually multiplex or maxplex cytokine kit. What you see in the picture is on microbeads which have bound anti-interleukins, so different of those molecules. There are 12 of them. When the cells are able to secrete, they are captured, and then we have another antibody with a different color that will detect this. So in this case, from one sample, one supernatant, you can analyze 12 of those different cytokines or factors or molecules. And I want to show you an example using the maxplex cytotoxic T and NKT cells. So in this case here, you can find out that if you incubate your NK cells with a target cells, NK cells will release some of cytokines. The 12, the ones that you can be detecting using this kit are listed on the left side. So it includes interleukin 2 and most important like granzyme B and perforin for NK cells. And with one sample, again, you can measure all of these, and you don't have to worry. You can just collect your samples, your supernatants, freeze them, and once you have enough samples, you run it, and you get all the information ready for you. Okay, so once you have determined how many or how much cytokines you have, you also want to know if your NK cells are able to kill. So what we do here, this is just an example. We have labeled our target cells with a dye, and in this case we use a cell trace, and you can see that you can count your cells when you dye them and you just run a normal FACS plot. If you incubate either with natural killer cells that are not transduced or with CAR NK cells, you will observe that at different effector to target ratios, the NK cells will kill the target cells. And you can quantify these by looking at the FACS plot. The less count, the less of the target cells remaining in your sample. So for example, in the effector target ratio 5:1, you have a couple of cells remaining, so NK cells are able to kill, But if you use CAR NK cells, they almost eliminate all of the target cells. If you want to know more about the protocol regarding these CAR NK cells, I'd invite you to download our publication. It's open access, so it's free for you to use, in which we have there step-by-step how to generate CAR NK cells using all of the tools and reagents that I just showed you. Moreover, if you are interested in how to cultivate NK cells or how to do a phenotypic analysis, you can always go to our application tab for NK cells. There's a lot of protocols there. You can download them, and you would also have this downloadable protocol with a timeline, the products and tips, and also some data. So now let's go to the last part. Very briefly, I just want to encourage you to always think translational. So I've shown you already all the tools that you can have to study NK cells on your research setting. So you can have from sample preparation to cell isolation, cytokines, medium, and recombinant antibodies. But if you want to take these to the next level, we also have all the tools that you need to actually study clinical or to do the practical clinical application of NK cells. I'm not going into too much details, but I do want to introduce your platform for this. This is the CliniMax Prodigy. It's an automated cell processing system for GMP compliant cell manufacturing. It allows you to not only isolate cells, but to culture or to do transduction, and you can start from the beginning using one single tubing set that is only one use in a closed system with an interface that allows you to program it to do different things. So for the NK cells, we're working on this, and we have two different processes. One of them is to isolate NK cells, the one on the left, CD3, CD56 system, and the other one we're working on them is our natural killer cell engineering system. And in this, both of them, it will allow you to start with leukapheresis products, isolate NK cells, and have your final NK cells in your infusion solution, for example, for infusion to a patient. Or in the case of the natural killer cell engineering, it will allow you to automatically isolate NK cells, do the transduction and expansion in one instrument. This is only one system, it's closed system, and then you will have at the end of 14 days your NK cells. So, okay, I'm getting to the end line. In general, I would just like to summarize by encourage you again to think translational right from the start. All the reagents, protocols, or the support throughout the whole NK or CAR NK cell workflow you have seen today, and they're available to you. We also have all these in max GMP quality for seamless transition from bench to the bedside. And once you discover the perfect protocol, of course, you can always translate into CliniMax Prodigy platform, which allows standardized and transferable and scalable obtention of NK cell products. So again, choose Miltenyi Biotec as your partner in CAR NK cell manufacturing or NK cell manufacturing. And if you are interested in getting to know more, make sure you visit our applications tab on NK cells. You will also find more webinars, application notes, workflow protocols, scientific posters. We're happy to share everything that we have with you. And with this is my last slide. I would like to thank you very much for your attention and for coming today.
Thank you, Martha, for your presentation. A quick reminder for our audience on how to submit questions. Simply type them into the drop-down box located on the far left of your presentation window labeled Ask a Question and click on the Send button. Martha will answer as many questions as time permits. All right, our first question is, will I have a problem if I use CD56 microbeads and I want to perform ADCC assay?
Nice to hear that question. I think I have it very often, and I am happy to say that we actually have data showing that using CD56 microbeads, of course, you will enrich NK and NKT cells, but we have to remember that this ADCC assay is based on the expression of CD16. And because NKT cells express very little or almost no CD16, they will not interfere with your ADCC assay. So you can be very confident of using all of the straight from products, CD56 microbeads, enrich your NK and NKT, and perform your ADCC without a problem.
Thank you. All right, your next question. What are the different formulations and formats of NK Max Medium?
Yes, so for the NK Max Medium, we're actually really proud of this media. It performs really, really nice. We have now a research version, so it's a bottle of 500 ml, but we have also the GMP grade for all the translational applications and clinical manufacturing, for example, on the Clinimax Prodigy. And this GMP version is available in a 2-liter flexible bags. And the key thing is that it has the same formulation as the research version. So when I tell you that it's truly translational, it is truly translational. Whatever you do on your research lab can be translated into your clinical protocol.
Now, does the NK Max Medium contain activating reagents or cytokines?
No, actually it doesn't. This NK Max Medium itself is chemically defined, does not contain any protein, no human serum albumin, no insulin, no cytokines. We did this so that each researcher can customize the desired cocktail or cytokines. You know, now there are a couple of different protocols. You can create memory-like NK cells. You can create mature NK cells. So it all depends on the cytokines that you put. So we do not have any cytokine inside the media. You are free to choose whatever your cocktail you want to choose.
Now, which cytokines do you recommend for the expansion of NK cells?
So for the expansion of NK cells, we started actually with a protocol using only interleukin-2, but a couple of publications have shown that the addition of interleukin-15 is actually really good. So our normal protocol includes interleukin-2 and 15, so we recommend that. This protocol, you can download it in our webpage actually, step by step, and it tells you with how many cells we start, when do we change the media, and you will get the results that we have also, that you've seen in the graphs today. But also, of course, you can use others like interleukin-12 or 18. Many other researchers are using this to create like memory-like NK cells. And one thing that I just want to highlight is that you make sure that you use the premium grade cytokines to make sure that you use the right dose and, of course, to have the best reproducibility later.
Great, thank you. Looks like we have time for one more question. Can you share the protocol for CAR NK cell generation?
Yes, we will have it available in our webpage. Right now it is not, but I am sure by the end of 2019 it will be there. So you just make sure that you go to our website and it will be updated there. We have an application tab with the NK cells. You just go there and you will probably find it.
I would like to once again thank Martha for her time today and her important research. I'd also like to thank the audience for joining us and for their interesting questions. Any questions we did not have time for today or those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. We would also like to thank Labroots and our sponsor Miltenyi Biotec for underwriting today's educational webcast. This webcast can be viewed on demand. Labroots will alert you when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's event. Until next time, goodbye.